Pyruvate is a key molecule in cellular metabolism. Pyruvate is a 3-carbon organic acid with a chemical formula of C3H3O3 mannans. Pyruvate serves as the end product of glycolysis, the first stage of cellular respiration. Pyruvate acts as a crucial metabolic junction, directing carbon flow through various pathways depending on cellular conditions. The fate of pyruvate varies based on oxygen availability, cellular energy demands, and other factors. Understanding pyruvate's fate is crucial for comprehending how cells generate and utilize energy under different circumstances. Pyruvate stands at the crossroads of metabolism, directing carbon flow based on cellular needs. Glycolysis is the universal pathway that breaks down glucose into pyruvate molecules. This process occurs in the cytoplasm of all cells and consists of 10 enzymatic reactions. Glycolysis is an ancient metabolic pathway that functions without requiring oxygen. During glycolysis, one glucose molecule containing 6 carbons is converted into 2 pyruvate molecules with 3 carbons each. The pathway can be divided into a preparatory phase and a payoff phase. Glycolysis generates energy in the form of ATP and NADH. For each glucose molecule, the process results in a net gain of 2 ATP molecules and 2 NH molecules. While 4 ATP are produced in total, 2 ATP are consumed in the early steps, giving a net gain of 2 ATP. Glycolysis is one of the most ancient metabolic pathways, dating back approximately 3.5 billion years. It functions with or without oxygen, serves as a gateway to multiple metabolic pathways, and is critical for energy production in all living cells. Glycolysis is a critical metabolic pathway that produces 3 essential products. These products are ATP, adenosine triphosphate, NADH, nicotinamide adenine dinucleotide, and pyruvate. ATP, or adenosine triphosphate, is the primary energy currency of the cell. It contains high-energy phosphate bonds that store and release energy when broken. Glycolysis produces a net gain of 2 ATP molecules for each glucose molecule processed. NADH, or nicotinamide adenine dinucleotide, is a high-energy electron carrier. During glycolysis, 2 NADH molecules are produced per glucose molecule. These electron carriers store energy and will later transfer it to other metabolic pathways. NADH is particularly important for cellular redox reactions and energy production in the electron transport chain. Pyruvate is the final carbon-containing product of glycolysis. Each glucose molecule produces 2 pyruvate molecules. As a 3-carbon molecule, pyruvate stands at a critical metabolic crossroads. Depending on cellular conditions and oxygen availability, pyruvate can enter various metabolic pathways for further energy production or biosynthesis. To summarize, glycolysis converts one glucose molecule into 2 ATP molecules, 2 NADH molecules, and 2 pyruvate molecules. These products are essential for cellular function, providing energy, maintaining redox balance, and serving as substrates for downstream metabolic pathways. Pyruvate stands at a critical metabolic crossroads in the cell. Its fate depends primarily on oxygen availability, but also on the cell's energy needs and metabolic state. Under aerobic conditions, pyruvate is converted to acetyl CoA, which enters the citric acid cycle. In low-oxygen environments, animal cells convert pyruvate to lactate through anaerobic glycolysis. Yeast and certain microorganisms transform pyruvate into ethanol through fermentation. Pyruvate can also enter various biosynthetic pathways, serving as a precursor for amino acids, lipids, and other essential molecules. This versatility makes pyruvate a central intermediate in cellular metabolism, capable of adapting to different cellular conditions and requirements. The balance of these metabolic pathways is tightly regulated to meet the cell's current needs, highlighting pyruvate's crucial role at this metabolic junction. In aerobic conditions, pyruvate follows a specific metabolic pathway. After glycolysis occurs in the cytoplasm, pyruvate must enter the mitochondria for further processing. Pyruvate molecules produced from glycolysis in the cytoplasm are transported across the mitochondrial membrane. Inside the mitochondria, pyruvate undergoes a complex transformation catalyzed by the pyruvate dehydrogenase complex. This enzyme complex requires NAD+ as a cofactor to accept electrons. The pyruvate molecule is decarboxylated, meaning a carbon dioxide molecule is removed. The remaining acetyl group is attached to coenzyme A, forming acetyl CoA. During this process, NAD+ is reduced to NADH, capturing the electrons released from pyruvate. The overall reaction can be summarized as pyruvate plus coenzyme A plus NAD+ yielding acetyl CoA, carbon dioxide, NADH, and a proton. The acetyl CoA produced in this reaction enters the citric acid cycle, linking glycolysis to the next stage of aerobic respiration. This aerobic conversion of pyruvate to acetyl CoA is crucial for efficient energy production. It requires oxygen, occurs in mitochondria, links glycolysis to the citric acid cycle, and produces one NADH molecule per pyruvate. The pyruvate dehydrogenase complex, often abbreviated as PDC, is a crucial multi-enzyme system in metabolism. This complex catalyzes the conversion of pyruvate, the end product of glycolysis, to acetyl CoA. The PDC is located in the mitochondrial matrix in eukaryotic cells, at the interface between glycolysis and the citric acid cycle. The complex consists of 3 primary enzymes working together. The complex also requires 5 essential coenzymes to function properly. The PDC catalyzes a complex multi-step reaction process. The reaction catalyzed by the PDC is irreversible and represents a committed step toward the complete oxidation of glucose-carbon atoms in the citric acid cycle. During this process, pyruvate is converted to acetyl CoA, releasing carbon dioxide and generating NADH for the electron transport chain. This crucial conversion provides the link between glycolysis and the citric acid cycle, enabling the complete oxidation of glucose. The citric acid cycle is a central metabolic pathway where acetyl CoA is completely oxidized. After pyruvate is converted to acetyl CoA by the pyruvate dehydrogenase complex, it enters this important aerobic pathway. The cycle begins when acetyl CoA combines with oxaloacetate to form citrate, a 6-carbon molecule. This initiates a series of enzymatic reactions that progress around the cycle, generating important products. Throughout the cycle, several key products are generated, which drive energy production in the cell. The cycle produces 3 NADH and 1 FADH2 molecules, which carry electrons to the electron transport chain. It also generates 1 GTP or ATP molecule directly through substrate-level phosphorylation. Most importantly, the cycle completely oxidizes the acetyl group, releasing 2 carbon dioxide molecules. By the end of the cycle, oxaloacetate is regenerated and ready to accept another acetyl CoA molecule, continuing this critical process of cellular respiration. Oxidative phosphorylation is the culmination of cellular respiration, where the majority of ATP is generated. The electron transport chain is located in the inner mitochondrial membrane. The electron transport chain consists of 4 protein complexes embedded in the membrane, plus ATP synthase. NADH and FADH2 produced during pyruvate oxidation carry high-energy electrons to the electron transport chain. As electrons flow through the chain, they release energy that is used to pump protons across the membrane. Complexes 1, 3, and 4 pump protons from the matrix to the intermembrane space, creating a proton gradient. The proton gradient drives ATP synthesis as protons flow back through ATP synthase. This process of oxidative phosphorylation generates the majority of ATP from glucose oxidation, with a total yield of about 30 to 32 ATP molecules per glucose molecule. Oxidative phosphorylation is the most efficient energy-producing pathway in aerobic organisms, capturing much more energy than anaerobic processes. The fate of pyruvate in aerobic conditions leads to a remarkable energy yield through complete oxidation. When oxygen is available, pyruvate enters the mitochondria and undergoes complete oxidation through the citric acid cycle and oxidative phosphorylation. Let's examine the ATP yield from the complete oxidation of one pyruvate molecule. During pyruvate dehydrogenase complex activity, 1 NADH is produced, which generates approximately 2.5 ATP through the electron transport chain. The citric acid cycle produces 3 NADH molecules, which yield about 7.5 ATP, 1 FADH2 yielding 1.5 ATP, and 1 GTP, which is equivalent to 1 ATP. Additionally, acetyl CoA entry into the citric acid cycle produces two carbon dioxide molecules, completing the oxidation process. In total, the complete oxidation of one pyruvate molecule generates approximately 15 ATP. Since one glucose molecule produces two pyruvate molecules through glycolysis, the complete aerobic oxidation of glucose yields approximately 30 ATP molecules. This demonstrates the remarkable efficiency of aerobic metabolism compared to anaerobic pathways, which produce only 2 ATP per glucose during glycolysis. This high energy yield is why aerobic metabolism is the preferred pathway when oxygen is available. Under anaerobic conditions, when oxygen levels are low in animal cells, pyruvate follows a different metabolic fate. Instead of entering the mitochondria for aerobic respiration, pyruvate is converted to lactate through a process catalyzed by the enzyme lactate dehydrogenase, or LDH. This reaction is critical because it regenerates NAD+ from NADH. During glycolysis, NAD+ is reduced to NADH, and this needs to be reversed for glycolysis to continue. By converting pyruvate to lactate, LDH facilitates the oxidation of NADH back to NAD+. This NAD+ can then return to glycolysis, allowing the process to continue generating ATP even when oxygen is unavailable. During intense exercise, when oxygen delivery to muscles cannot meet demand, lactate formation increases dramatically. As lactate accumulates in muscle cells, it causes several physiological effects. First, lactate contributes to metabolic acidosis by releasing protons, which lowers the pH in muscle tissue. This acidosis leads to decreased muscle contractility, pain, and impaired enzyme function, all contributing to muscle fatigue. This is why high-intensity exercise cannot be sustained for long periods. To summarize, the anaerobic fate of pyruvate in animal cells involves its conversion to lactate. This process regenerates NAD+, allowing glycolysis to continue producing ATP even without oxygen. However, the resulting lactate accumulation and acidosis contribute to muscle fatigue during intense exercise. In yeast cells under anaerobic conditions, pyruvate undergoes a different fate than in animal cells. This process, known as alcoholic fermentation, converts pyruvate to ethanol through a two-step pathway. In the first step, the enzyme pyruvate decarboxylase removes carbon dioxide from pyruvate, converting it to acetaldehyde. This reaction requires thiamine pyrophosphate as a cofactor. The carbon dioxide released during this step creates the bubbles in bread dough, causing it to rise during baking. In the second step, alcohol dehydrogenase reduces acetaldehyde to ethanol, using NADH as an electron donor. This regenerates NAD+, which is essential for glycolysis to continue. This regeneration of NAD+ is crucial because it allows glycolysis to continue producing ATP under anaerobic conditions, even when the electron transport chain is not functional. Alcoholic fermentation is the basis for two important food processes: bread making, where carbon dioxide causes dough to rise, and the production of alcoholic beverages, where ethanol is the desired product. To summarize, alcoholic fermentation is a two-step process that converts pyruvate to ethanol, regenerating NAD+ to sustain glycolysis under anaerobic conditions. This pathway produces both carbon dioxide and ethanol as byproducts, which are utilized in bread making and alcoholic beverage production. NAD+ regeneration is a crucial process that allows cells to maintain energy production in anaerobic conditions. At the core of anaerobic metabolism is the NAD-NADH cycle. During glycolysis, NAD+ accepts electrons to form NADH. In aerobic conditions, these electrons would enter the electron transport chain. But in anaerobic conditions, fermentation processes regenerate NAD+ from NADH. Let's see how this works in practice. Glycolysis produces pyruvate along with NADH. Without oxygen, cells need alternative ways to regenerate NAD+. In animal cells, pyruvate is converted to lactate, regenerating NAD+ in the process. In yeast cells, pyruvate undergoes alcoholic fermentation to produce ethanol and carbon dioxide, also regenerating NAD+. This NAD+ regeneration is critical because it allows glycolysis to continue producing ATP even when oxygen is unavailable. Let's summarize why NAD+ regeneration is so important in anaerobic conditions. Oxygen availability is the primary factor that determines the metabolic fate of pyruvate. To understand this relationship, let's look at how oxygen affects pyruvate metabolism within the cell. In the presence of oxygen, pyruvate enters the mitochondria for complete oxidation. Inside the mitochondria, pyruvate is converted to acetyl CoA, which enters the citric acid cycle. Electrons then flow through the electron transport chain, which requires oxygen as the final electron acceptor. Without oxygen, pyruvate remains in the cytoplasm and undergoes fermentation. In animal cells, pyruvate is converted to lactate, while in yeast cells it's converted to ethanol and carbon dioxide. Essentially, oxygen availability acts as a metabolic switch, determining whether energy production will be efficient through complete oxidation or limited through fermentation. Enzymes controlling pyruvate's fate are tightly regulated to ensure appropriate metabolic responses. The fate of pyruvate is primarily controlled by two key enzymes, pyruvate dehydrogenase, or PDH, and lactate dehydrogenase, or LDH. Pyruvate dehydrogenase, or PDH, is inhibited by high levels of ATP, NADH, and acetyl CoA. When these energy molecules decrease, PDH is activated, directing pyruvate toward the aerobic pathway for ATP production. Lactate dehydrogenase activity, on the other hand, increases during oxygen limitation. When oxygen levels decrease, LDH activity increases to convert pyruvate to lactate, which regenerates NAD+ needed for glycolysis to continue. These enzymatic regulatory mechanisms ensure appropriate metabolic responses to changing cellular conditions. PDH inhibition conserves glucose when energy is abundant, while activation of PDH drives aerobic metabolism when energy is needed. LDH activation during low oxygen helps maintain glycolysis, allowing cells to adapt quickly to changing conditions. Nutritional status significantly influences the metabolic fate of pyruvate in the cell. Pyruvate's metabolic fate depends on whether the body is in a fasting or fed state. During fasting, pyruvate is primarily directed toward gluconeogenesis, the production of new glucose. This process is essential for maintaining adequate blood glucose levels to support vital functions, especially for the brain. After meals, especially those rich in carbohydrates, pyruvate follows different metabolic paths. Pyruvate can be oxidized through the citric acid cycle to generate energy in the form of ATP. Alternatively, when energy needs are met, pyruvate can be directed toward fatty acid synthesis for energy storage. The balance between these pathways is regulated based on the body's energy status. When energy levels are high, more pyruvate is directed toward fatty acid synthesis for storage. Conversely, during increased energy demand, pyruvate is primarily used for ATP production through oxidative phosphorylation. Thus, the cell's nutritional status plays a critical role in determining whether pyruvate is used for gluconeogenesis during fasting or for energy production and storage after meals. Hormones play a crucial role in regulating pyruvate metabolism, coordinating how cells process this key metabolite. Insulin, released after meals when blood glucose is high, promotes the oxidation of pyruvate through the pyruvate dehydrogenase complex. Insulin also enhances the conversion of pyruvate to fatty acids for energy storage, especially in adipose tissue. In contrast, glucagon and epinephrine are released during fasting or stress conditions. These counter-regulatory hormones stimulate gluconeogenesis, converting pyruvate back to glucose to maintain blood glucose levels. Hormones coordinate pyruvate metabolism across different tissues to maintain whole-body energy balance. In the liver, insulin suppresses gluconeogenesis from pyruvate, while glucagon stimulates it. In muscle, insulin promotes glucose uptake and pyruvate oxidation, while epinephrine shifts metabolism toward glycogen breakdown. In adipose tissue, insulin enhances the conversion of pyruvate-derived acetyl CoA to fatty acids, while epinephrine stimulates fat breakdown. In summary, hormones act as master regulators of pyruvate metabolism, directing this key metabolite toward different fates based on the body's energy needs. The balance between insulin, glucagon, and epinephrine ensures that pyruvate is appropriately directed to maintain energy homeostasis under varying physiological conditions. Cellular energy demand directly influences how pyruvate is utilized in metabolic pathways. In high energy demanding states, such as during exercise or rapid cell growth, pyruvate is preferentially oxidized via the citric acid cycle. This aerobic pathway maximizes ATP production, generating up to 32 ATP molecules per glucose molecule through oxidative phosphorylation. Conversely, when energy demands are low, but biosynthetic needs are high, pyruvate is directed toward anabolic pathways. In these conditions, pyruvate can be diverted to fatty acid synthesis, particularly when carbohydrates are abundant and energy storage is beneficial. Pyruvate can also serve as a precursor for amino acid synthesis, particularly for alanine, through a simple transamination reaction. The balance between energy production and biosynthesis is carefully regulated. This metabolic flexibility allows cells to adapt to changing physiological conditions and nutrient availability. Genetic and molecular factors play a crucial role in determining how cells process pyruvate. Genetic factors affect pyruvate metabolism primarily through the expression levels of key enzymes. Transcriptional regulation responds to various signals, including nutrients, oxygen levels, and hormones. Several key enzymes are subject to this genetic regulation, including pyruvate dehydrogenase kinase, lactate dehydrogenase, and pyruvate carboxylase. The expression levels of these enzymes shift the metabolic balance between aerobic and anaerobic pathways. Mutations in these metabolic enzymes can cause serious disorders that affect how cells utilize pyruvate. Environmental factors significantly impact pyruvate metabolism pathways. pH is a critical factor that determines which metabolic pathways are favored. The pH scale ranges from acidic to alkaline. In acidic conditions, with pH below 7, cells favor lactate formation and anaerobic pathways. This is commonly seen during intense exercise when muscle tissue becomes more acidic. In alkaline conditions, with pH above 7, pyruvate oxidation and aerobic metabolism are enhanced, resulting in more efficient energy production through the citric acid cycle. Temperature significantly affects enzyme kinetics in pyruvate metabolism. Enzymes have an optimal temperature range where they function most efficiently. As temperature increases from cold to body temperature, enzyme activity increases, accelerating pyruvate metabolism. The optimal temperature for human enzymes is around 37 degrees Celsius. However, at higher temperatures, enzymes begin to denature, losing their structure and function. This dramatically reduces pyruvate metabolism efficiency. Several cofactors are essential for proper pyruvate metabolism. These molecules assist enzymes in catalyzing reactions. Na+ functions as an electron acceptor in redox reactions, playing a crucial role in both aerobic and anaerobic pyruvate metabolism. Coenzyme A, or CoA, serves as a carrier for acyl groups, enabling pyruvate to enter the citric acid cycle as acetyl CoA. Thiamine pyrophosphate, derived from vitamin B1, is essential for the pyruvate dehydrogenase complex that converts pyruvate to acetyl CoA. The combined effects of environmental factors determine which metabolic pathway pyruvate will follow. Alkaline pH, optimal temperature around 37 degrees, and available cofactors like NAD+, CoA, and thiamine pyrophosphate promote the aerobic pathway, converting pyruvate to acetyl CoA. Conversely, acidic pH, temperature extremes, and limited cofactor availability favor anaerobic metabolism, converting pyruvate to lactate. Pyruvate serves as a primary substrate for gluconeogenesis, the process of generating glucose from non-carbohydrate sources. Gluconeogenesis primarily occurs in the liver and kidneys, where specialized enzymes catalyze this multi-step pathway. The pathway begins with pyruvate, which is carboxylated to form oxaloacetate by the enzyme pyruvate carboxylase. This reaction requires ATP as an energy source. Oxaloacetate then undergoes a series of enzymatic reactions, including decarboxylation, reduction, and dephosphorylation steps, eventually forming glucose. This pathway becomes crucial during fasting states when blood glucose levels need to be maintained despite depleted glycogen stores. During fasting, blood glucose levels must be maintained within a narrow range to support brain function. As liver glycogen stores are depleted, pyruvate derived from amino acids becomes a critical source for glucose production. The rate of gluconeogenesis increases significantly to maintain glucose homeostasis, preventing hypoglycemia during prolonged fasting periods. Pyruvate serves as an important precursor for fatty acid synthesis, providing a pathway to store excess energy. This process begins in the mitochondria, where pyruvate is first converted to acetyl CoA. The acetyl CoA must then be transported from the mitochondria to the cytoplasm, where fatty acid synthesis takes place. In the cytoplasm, acetyl CoA is converted to malonyl CoA by the enzyme acetyl CoA carboxylase, or ACC. This is a rate-limiting step in fatty acid synthesis. Malonyl CoA then enters the fatty acid synthase complex, where it's used to build fatty acid chains through sequential addition of 2 carbon units. This pathway is particularly active after carbohydrate-rich meals when energy is abundant. High insulin levels activate the ACC enzyme, promoting the conversion of excess carbohydrates into stored fat. This metabolic pathway represents an important mechanism for energy storage, allowing excess carbon to be preserved as fat for future use when energy demands exceed intake. Pyruvate plays a crucial role in amino acid synthesis, serving as a key precursor for several amino acids. The most direct pathway is the conversion of pyruvate to alanine through a process called transamination. During transamination, an amino group is transferred from another amino acid to pyruvate, forming alanine. This is a reversible reaction catalyzed by alanine aminotransferase. Pyruvate can also be carboxylated to form oxaloacetate, which serves as a precursor for aspartate and asparagine. Oxaloacetate undergoes transamination to form aspartate. Aspartate can be further modified through amidation to form asparagine. Pyruvate can also enter the TCA cycle, where it contributes to the formation of alpha-ketoglutarate. Alpha-ketoglutarate is transaminated to form glutamate, another important amino acid. Glutamate can be further modified through amidation to form glutamine. Glutamate can also be converted to proline through a cyclization process. Through these pathways, pyruvate acts as a critical link between carbohydrate metabolism and protein synthesis. In summary, pyruvate contributes to the synthesis of multiple amino acids: alanine through direct transamination, aspartate and asparagine via oxaloacetate, and glutamate, glutamine, and proline via alpha-ketoglutarate. This metabolic versatility makes pyruvate a crucial connector between carbohydrate metabolism and amino acid synthesis, highlighting its central role in cellular metabolism. In this final section, we'll summarize the central role of pyruvate in cellular metabolism. Pyruvate stands at a critical crossroads in metabolism, serving as a central hub that connects multiple metabolic pathways. Under aerobic conditions, pyruvate is oxidized to acetyl CoA, entering the citric acid cycle and eventually leading to substantial ATP production through oxidative phosphorylation. In animal cells, under anaerobic conditions, pyruvate is reduced to lactate, allowing for continued glycolysis by regenerating NAD+. In yeast and some microorganisms, pyruvate undergoes alcoholic fermentation to produce ethanol, also regenerating NAD+. Additionally, pyruvate serves as a precursor for various biosynthetic pathways, including gluconeogenesis, fatty acid synthesis, and amino acid production. The fate of pyruvate is tightly regulated by several key factors. Oxygen availability determines whether pyruvate follows aerobic or anaerobic pathways. Cellular energy demands influence whether pyruvate is catabolized for energy or diverted to biosynthetic processes. Nutrient status, including the availability of glucose and other substrates, affects pyruvate utilization. Hormonal signals coordinate pyruvate metabolism across tissues to maintain whole-body energy homeostasis. This precise regulation of pyruvate metabolism allows cells to adapt to changing environmental conditions and metabolic demands. Understanding the central role of pyruvate provides insight into how cells maintain energy homeostasis and respond to metabolic challenges, underlining its significance in cellular metabolism.